quite a lot about Moderna and Pfizer vaccination, but in South
Mhm.
African reality, and most of our viewers will be located in South Africa,
Yes.
we cannot get Moderna or Pfizer, and not because of the costs, but simply because it's not feasible. Why is it not feasible, and why AstraZeneca option is better for African countries?
Okay. So RNA is quite unstable. What's in the mRNA vaccines, obviously. So they have to be stored at really cold temperatures so that the vaccine doesn't degrade. And so the Pfizer vaccine has to be stored at around minus 70 degrees Celsius, and the Moderna vaccine has to be stored at around minus 20 degrees Celsius. And obviously, in South Africa, that's not really a reality for every clinic or hospital that needs to vaccinate people. So the fridges and freezers may be available, but space-wise, we don't have enough space to store all the vaccines that we need to vaccinate everyone. So in our case, we are procuring a vaccine that can be stored at room temperature, so it's stable. Not at room temperature, my apologies. We are procuring a vaccine that is stable at regular fridge temperature, so 2 to 8 degrees Celsius.
So how does it? So in South Africa, because we are procuring AstraZeneca vaccination, how does it differ from Moderna and Pfizer that it can be stored at a standard fridge temperature?
Okay, so how it differs is we use what we call a viral vector. So we take another virus. In the case of the AstraZeneca vaccine, it's adenovirus 5. And basically, we empty out. So remember, I told you that viruses have a protein shell and then the genetic material is on the inside. What we do is we take out that genetic material of the adenovirus and we're left with a shell. And then we take that RNA of the spike protein or the S protein of SARS-CoV-2 and we put that into the adenovirus. And basically, what happens is once that gets into your cell, your cells can start producing spike protein.
So you're taking the DNA of the virus.
The genetic material of SARS-CoV-2 and putting it into the adenovirus, yes. So it's not infectious. It won't cause any kind of illness. And so the adenovirus is basically just a shell. In this case, it's functioning similarly to the lipid nanoparticle in the mRNA-based vaccine.
So it's basically kind of like a box from Takealot. You order something and then Takealot sends the massive box, but inside is
what you want. We've got your spike protein. Basically, that's what it does.
And then you make your own product, and that's not
so your cells. Yes, non-infectious. So in the case of all three of these vaccines that we've discussed now, the Pfizer, the Moderna, and the AstraZeneca vaccines, it's your body that's making these proteins. So all we're doing is we're giving it an instruction to say, okay, this is what the protein must look like, and then your cells will produce it. And then because your body recognizes it as, oh, but I've never seen this protein before, it's not mine, your immune system will go in and say, okay, we need to learn to recognize this so that when we encounter it again, we can just smack it down. Basically, that's how vaccination works.
And one thing about the vaccination, so recently there's been a massive fear about the new variants, the UK variant and South African variant of SARS-CoV-2. Can you tell us if the vaccination work against those new viruses?
So just to give a little bit of background, viruses evolve. It's part of their existence. They have to evolve to survive. And so it's not surprising that the virus is evolving, but it is encouraging that it's evolving very slowly over time. So the first cases of COVID we saw were in December 2019, and the biggest notable changes in variants that could be potentially worrying were described last year, so the UK and the South African variants. And so in terms of, you're just going to have to trust me on this one, in terms of viral evolution, that is very slow. And so basically when they looked at these variants, we can sequence the proteins, and we can begin to understand a little bit about how it has changed as it evolved, and potentially what function these mutations confer to the virus. And the South African variants were both causing a bit of a stir because they were worried. Sorry, okay, I'm back. Because scientists were worried that it may have implications for treatment, vaccine design, because we know that these variants are more transmissible than previous variants. And so Pfizer has come out with some preliminary data to show that their vaccine is effective, well, will be effective against the South African and the UK variant. And it's looking hopeful, because then it's very likely that other vaccines will also be successful, because they're all designed with the same target, the spike protein, as they target. So we don't have all of the answers yet, but I wouldn't start to panic.
And those two variants, the new variants, they seem to be more infectious. Why is that?
So basically, the most notable mutations that occurred were in the spike protein, or in the S protein. And we've been speaking about this protein a lot, but if you don't know, the function of the spike protein is to help the virus enter cells. So that's the protein that recognizes your cells, and then is able to enter our bodies through that, through interactions between our cells and that spike protein. And so what happened with the UK and the SA variants is they have mutated in the spike protein. And so basically these mutations allow it to enter cells a little bit more efficiently, transmit a little bit more efficiently. And so, yeah, that's why we're seeing higher transmission. Studies with these new variants are ongoing. Like I said, we need to determine exactly how resistant it might be to neutralization by antibodies. But as I said, there's some preliminary evidence from Pfizer already to show that they are sensitive to antibodies that are produced as a result of being vaccinated. So still quite a few questions that remain, but that's the gist of things.
But there is a hope, luckily.
There's hope. Oh no, there's most definitely hope. Most definitely. And as I said, it's evolving slowly. So we don't. I don't think in the near future we really have to worry too much. The SA and the UK variants are quite similar, and so it shows that potentially these mutations have a benefit for the virus because they both evolve the same kind of mutation. And so yeah, it is becoming a little bit more, a little bit better at transmitting. But I mean, there's no reason why, if you are already masking up, washing your hands, maintaining physical distance, not gathering indoors, that it should be more of a risk to you personally.
And since you're a specialist on HIV, there has been questions why we are so quick with developing the vaccination against this newly developed virus, the virus that just infected us a couple months ago, but we still haven't found anything for HIV. Why is that?
So I mentioned earlier about mutation rates. So SARS-CoV-2 mutates very slowly. In the case of HIV, unfortunately, mutates really quickly. And that's a problem because as you see with we're having to test the vaccine, antibodies that people produce when they've been vaccinated. We have to test those antibodies to see if they'll be effective against all the different kinds of variants. In the same way, you have to do that with HIV, and within any given person, there are multiple different variants of HIV. So people living with HIV have multiple variants circulating. And so it becomes difficult to even treat or even, I guess, vaccinate a whole bunch of people when you have so many different variants that are transmitting at the same time. I hope that makes sense. Sorry. It's kind of
No, it does. It's important to emphasize the evolution, that it's viruses are the best example of evolution.
Yeah. So if you have a more diverse population of viruses, it's less easy to neutralize them because they look so different to each other, and even your own immune system struggles with that. So in natural infection with HIV, the immune system struggles to keep the virus in check because it's constantly evolving away from your immune system being able to recognize it. And that's why it's even been so difficult to kind of treat with anything other than antiretrovirals. So those are very effective, obviously. I said in terms of vaccination, it's a little bit more tricky than actually treating patients for HIV. So in the case of treatment for HIV is very effective, and preventing spread of HIV is very effective. And so currently we rely on those methods for controlling HIV infection, as opposed to SARS-CoV-2, where we don't really have many treatments, especially not in many places in the world. And it spreads, it's a little bit more difficult to control the spread because it's a respiratory virus. And so a vaccine for this, because of the fact that it's so similar, doesn't change very frequently, a vaccine will be most effective.
So we've been talking about difficulties with HIV vaccination versus COVID, but also there have been a lot of questions why the vaccination came so quick. What happened there?
So coronaviruses aren't new viruses in themselves. Coronaviruses have existed for a very long time and have been studied for a very long time. And so just because there's a new strain of coronavirus now, it doesn't mean that we haven't been studying coronaviruses previously. So we do know a little bit about them. Well, quite a bit about them. Some of you will remember the SARS epidemic and MERS epidemic, or outbreaks that happened a couple of years ago, and those were similar viruses. They were also coronaviruses. And so basically, every time there's an outbreak of something, we study it and then try to figure out, okay, if this ever happens again, how can we prevent this from happening again? So that research has been happening, and basically this time that there was an outbreak, we already had research on pandemic coronaviruses. And so the reason why this virus was renamed SARS-CoV-2 is because it's similar to the human coronavirus SARS-1. So yes, we had some research that was sitting on ice, and we were able to go and compare the sequence of this virus, SARS-CoV-2, with previous human coronavirus sequences, see how similar they were, test out the vaccines and therapeutics that we were testing out against those human coronaviruses in tissue culture. So we do these experiments in the lab to see if any of the existing treatments would work against this novel coronavirus. So basically there was research to kind of bounce off of when we discovered that this was another human coronavirus. And so that enabled us to get through um, through some of the initial barriers, the initial stages of research quite quickly. And then also because it was such a big threat, I mean, it's a global pandemic. Most countries in the world are struggling with this right now. So it's kind of an emergency. We need to find something to combat this. And so because it has everyone's attention and because everyone kind of wants to get rid of it, I'm not even going to mince words, they basically made funding available. They made, labs available. They made a whole bunch of resources available that usually we have to, we spend a lot of time trying to find for research. And because this was already available, people just snatched up the opportunities. And because it was also so urgent, basically a lot of the time that was taken for administrative stuff on projects, it was prioritized. So SARS-CoV-2 projects were pushed to the front of the queue for, assessment and stuff like that. So a lot of the admin around running trials and stuff like that happened a lot quicker than what it does in the normal, non-COVID era.
So I'll just stop you here. So you're saying that with the clinical trials, the things were pushed forward, but it doesn't mean that the trials had less safety precautions, right?
No, not at all. So the first thing is we can't rush enrolling participants. So participants will enroll whenever they come into the clinic or whenever they consent to being a part of the trial. And you cannot stop enrollment early, so you need to enroll enough people into a trial, and that will take out how much time it takes. I'll say that again. In order to study the immune responses in people who have been vaccinated, we have to wait for the immune response to respond, and then we can measure the responses, and that takes time. You can't rush that. So there's no possible way you can rush the clinical stuff. It's mostly just the administrative side of things that can be expedited.
So people who are scared that the vaccination came so quickly, they can relax now. All the safety precautions were taken.
Yes, they definitely relax. Yes.
So it is safe.
It's safe, yes. The safety data is available. There's quite a few places, and I'm sure you can link some of the articles. I don't want to be forward, but there's science websites that have basically deconstructed what's in the journals, so the publications publishing safety data and stuff like that. So you don't have to go and read the paper itself, not unless you want to. But they basically put it in simple layman's terms that you can understand, and the safety and efficacy data is out there for all of us to read. So I would encourage you to read it, read up on it, read up on how the vaccine works, and set yourself like your own mind at ease about these things. Because after all, we do worry about the safety of our parents, the safety of our friends, the safety of ourselves when we take these vaccine, then that is important. So yeah, but they are safe.
Okay, yeah, I will definitely include the links for anyone who's interested to nerd more a bit the vaccination, or is more worried about the vaccination, or wants to spread the word about it. So we're gonna have the links at below this video. So to summarize this conversation, for which I'm really grateful, Sherazaan, I think it was very informative. South Africa mostly will purchase at the moment AstraZeneca vaccinations, which can be stored in a standard fridge. Therefore, it's easier to distribute. And the vaccination mix contains mostly the viral genetic material that will be only a small particle of the virus will be produced by our own cells, so no infection itself with the virus. And we will not have many adverse effects of the vaccination, mostly a bit getting sore, from it, like a standard immune response. And if someone is prone to allergy, they should inform the administering nurse or doctor, because they might experience some more severe adverse effects. But
Mm.
the cases were only few, but the vaccinations were in thousands. So the rate is quite
Vaccinations are in millions already. So
already in millions.
yeah, there's I think it's about 6 million people have been vaccinated already, and the adverse allergic reactions that have been reported, like I said, are in the tens, so fewer than 100. So that's
Yes.
fewer than 100 in 6 million, which is minuscule if you think about.
Absolutely. No, the odds are much better if you take the vaccination and get the chance of having allergic reaction than if you actually decide to expose yourself to the virus naturally
Yeah,
and see what happens.
considering you may not know what kind of disease severity you'll have if you are exposed to the virus and what kind of long-term effects that may have, it's definitely better to get vaccinated.
No, absolutely. And I think it's also important to point out that despite the fact that initially it has been thought that only people that are older have severe effects of the natural infection, the younger people start getting quite severe reactions and they die. Even people are in their 30s. So it shows how complex is the immune system, and you might not know your underlying minor immunological deficiency that on daily basis is not visible, but it might appear to be very significant when you get infected with SARS-CoV-2.
Yes, and obviously there's a lot of these questions popping up now with the new variant that unfortunately I don't think we have definitive answers for at the moment. So is this new variant associated with more severe disease? Is it associated with more severe disease in younger people? And the research is ongoing. So I think the take-home message is just, if you can get vaccinated sooner rather than later. But in all ways, just try to protect yourself from acquiring SARS-CoV-2 in the first place, because it is very difficult to predict what your outcome is going to be. And even then, you don't want to be one of the people who is like spreading the virus around. So it's just better to kind of be cautious.
Now, be cautious, because even if we stick to our bubbles, turns out our small bubbles that we think of are not that small.
Yeah.
So that's also the problem. Well, thank you so much, Sherazaan, again, for your information, for your knowledge that you decided to share with me and our viewers.
Thank you. I hope it was helpful. And yeah, I can't wait for that vaccine, eh?
Absolutely.